Thank you. So I want to play it safe, and I want to open the panel to the audience first, because it's such a heterogeneous panel. I don't really have questions, too many questions to ask them. So, yes, please.
I agree, an excellent panel. My question's for Dr. Ross. I'm a clinician. Your implication of microneedling exosomes and it going to the brain is very exciting. What are you going to delve into? Can you extrapolate on my thought of really using it for anti-aging, and you were microneedling a face and it went to the brain?
Right. I, I haven't done, well, I can't do that exactly, but what I can do.
Duncan, can you use the mic?
Can you hear me? Oh,
Yes.
wow. I suppose I could do an animal model of that and try to track that, but in humans, what I'm planning on is epigenetic aging testing prior to those kind of treatments and try to just go with that. But also we're researching focused ultrasound and targeting certain parts of the brain to increase the blood-brain barrier permeability, and actually I've done those rat studies, and this would have been a good place to put those slides up, but I did show that we increased the permeability of that barrier with that focused ultrasound. So now, if you do a functional MRI and you know what you're targeting specifically, then I think that that's a therapy in the future. But it's just recently that I've thought about that.
I have a couple of questions for Brian. Brian, since you have moved to Singapore, Singapore has become the hotbed of all clinical research related to longevity. So what is happening in Singapore that allows, or from a regulatory perspective, or from a facilitating perspective that's happening right in Singapore, a model that other countries could emulate?
Yeah, I don't know if it's so much the regulatory perspective. I mean, I think that the regulations are very similar there to what we would do in the U.S. And you know, the interventions we're doing are in a largely healthy population, so it's actually fairly low risk. On the other hand, it can be scary to ethics boards when you're working with healthy people. So, you know, we've gotten AKG approved, for instance, to start with, and that's about the safest thing you could imagine, and that took 6 or 7 months of debating with the ethics board. So I don't think it's all that different. I think what's different is that there are resources there for us to test, and maybe we're moving relatively quickly because we're not taking the approach of doing a study that the FDA will immediately recognize. We're taking more of a data approach where we want to test many kinds of interventions in RCT-like studies where we can figure out what we think is really happening and then maybe design the ultimate study down the road. And so I think there's certainly resources that have been provided to us from the university, from philanthropy, we're working with companies. And I think there's a mindset there that this is the right thing to do. And so that's all great. And we're just trying to keep enough momentum to keep the studies going and keep the balls moving. Keep the balls in the air, I guess, is the right metaphor I'm looking for.
Yeah, because in the previous panel, we did talk about the lack of money, availability of money in terms of resources. So we had a lengthy discussion. So how did you get to a position that the resources are available to do all that you want to do, which the researchers here in the US are finding it extremely difficult? Is that the governmental support?
Well, there are people doing the kinds of clinical studies that we talked about, or at least thinking about them in the US. So I don't think we're the only ones doing them. I think that when you look at the TAME trial, it's just the cost is like massively higher for that study. And so that's going to make it would be very hard for me to do that in Singapore. I mean, there's no way I could recruit 3,000 people and have the resources to do that there. So I think the goals are a bit different comparing the smaller stuff we're doing to the bigger TAME studies. But there's certainly people thinking about smaller scale studies with geroprotectors around the world. And some of that's targeted more to disease. And I think there's also a bit of a mindset here to pick chronic conditions that are informative with respect to aging and linked to the pathways that the geroprotector targets. which is, as I said in my talk, I don't think there's a right or wrong approach right now. I don't think we should be
Sure.
all getting together and choosing one path at the moment. I think we need to explore different paths until we get a better feel for, how aging biomarkers are going to respond in what people with what interventions and how that relates to pathology and physiology, and then maybe come back in a few years and decide what the best strategy is. But yeah, I mean, the magnitude of the TAME study is, I think, the limitation. I think a lot of people realize the importance of it, and it's just hard. It's not it's a cheap drug off patent and a very expensive study.
Sure. And you talked about the lipid clock. Yeah. So, yeah. So, can you hear?
Yeah.
Okay. Sorry, apologize. You talked about the lipid clock, lipid-based clock.
Yeah.
And you also talked about the epigenetic clock having been used in one of your studies. So my question is does it make sense to put all the omics measurement together to create some kind of a composite measure of biological age rather than using any of these clocks in isolation?
Yeah, I think there's a motivation to try to put is that better? Sorry. My jet lag is wearing me down. I think there's a motivation to try to put the different clocks together and try to create some composite, but right now the challenge is there haven't been enough studies with multiple clocks. And so we don't know what's redundant and what's unique and how to put things together yet. And so that's one of the reasons I think we're measuring 8 different clocks in our intervention studies, and part of that's to understand how they fit together with each other and not only how they respond to the interventions. Ultimately, it may be a composite like that works best, yeah.
Thank you. So,
Well, I mean, I'm cheating a little bit because once you do the methylation, you can run all the methylation clocks as long as you have the sites. So, but we're using 4 primarily, but we have the potential to do more than that. We're looking at proteomics, lipidomics. There are also a couple clocks you can get from complete blood count parameters. And we're using a facial clock that does a 3D reconstruction of a person's face and estimates their biologic age based on that reconstruction. And that's only been done in the Chinese population, so we're using it in our Chinese population, but it needs to be developed. It's going to be ethnic specific. So.
There's quantific here?
you also talked
Quantific
about ethnicity and the impact the ethnicity has on the results or outcomes of these clocks. Now, here in the US, everybody, irrespective of what the ethnicity is, are using epigenetic clock and the outcome as the measure of biological age. So, what needs to change?
I missed the first part of that in the U.S.
So
The U.S. part I missed.
ethnicity and its impact on epigenetic clock. I think during your talk,
Yeah.
you mentioned that it needs to be trained specifically for different ethnicities. Right now, there is one epigenetic clock that we all use here.
Yeah, and it really hasn't been tested in a wide enough part of the world, and that's kind of what we're doing. And so it could be that different clocks work better than others for different ethnicities, the ones that already exist, but most all of them have been trained on Caucasian or
Yes.
Caucasian and African populations. And so when you're looking at applying those clocks in Indian and Malay populations, they don't work as well, at least in our hands so far. So they may have to be modified to get
Retrained,
that. I
yeah.
think also one of the things, I can't talk in too much detail about this because it's not published and it's a big consortium, but we did 4 methylation clocks in 10,000 Singaporeans. And interestingly, one of them, we also looked at telomeres, and one of the clocks correlates quite well with telomeres and the other 3 don't. And so I think that comes back to the notion that these clocks are all trying to measure aging, but they're tilted toward different hallmarks or pathways or aspects of aging, maybe unintentionally by the AI. And we may find as we develop, and this one thing we're doing is trying to measure each hallmark and pathway of aging too. And we may find that some clocks are more inflammatory and another clock might be more metabolic and one is more linked to telomeres. And I think we need to sort those things out as we go forward.
Thank you. Duncan, the question we discussed with regards to exosomes, I think yesterday because we had somebody representing from Sartorius here, and he was of the opinion that we have the right technology to package the kind of cargo that we want to package in exosomes and use it in vivo. Based on my research, that I don't really see a reliable technology available to package the cargo. What's your opinion on that?
Well, there have been studies of adding, say, a microRNA to the culture, and then that will be taken up by exosomes. But we haven't gotten to testing that yet. I think with the RNAs, you will end up with some antigenicity, just like the Moderna vaccines, et cetera. But I think it's absolutely possible. I mean, so packaging, as well as helping target that exosome via ligated proteins on the surface, is coming. I mean, I'm working on it. So I think we'll get there pretty soon.
Okay. And you use, for clinical applications, you do exosome purification exosome purification in larger quantities. So is that do you have methodologies that will take care of the heterogeneity that exists in an exosome population?
Well, that's a good question. I mean, if you look at that image from the D storm, that's a very, very heterogeneous population, which is kind of why I know it sounds heretical, but I don't care about exosomes. I care about therapeutic outcomes and then the amount of RNA in that solution, the amount of proteins in that solution, and that's going to correlate to a number of exosomes, yes, but does that really matter? And I think what has happened with everyone, I mean, even the FDA said, why don't you make your release criteria the particle count? And I'm like, no, we're not going to do that, you know? And I had to argue that point. And these clinical exosome products that are out on the market, they say, oh, I have a billion, I have a trillion. I mean, you know, I actually have 2.4 quadrillion particles if you want to go that route, you know, but that's not going to help. But it's an ongoing question. And right now I'm just going to stick with, that's why I created that unit. And I think that that's an effective way to do it. I haven't heard anything else like that. If somebody else would like to clarify it for me, I would appreciate it. But I think that a unit, just like Botox is sold in a unit, is a more appropriate method.
And if you were to use a zeta view, you will get a different number. If you use a nanosite, you will get... Okay, so if you use a zeta view, you will get a different number. If you use a nanosite, you will get a different number. Depending on what you're using to quantitate, you have a wide variety of numbers that you get on the same batch of exosomes.
Well, you can, right, the ExoView is a pretty good product, but again, it's showing you the number, it's just showing you the number of exosomes, and they do that by binding the exosome. A nanosite, for instance, say, well, when they sold it to me, you can have a 488 laser on it. Okay, great, well then I could put some of these markers and I could quantify. Well, then we found out that, oh, it's not powerful enough with these exosomes moving around to actually see it. So they had never actually done that when they sold it to me with that idea, you know. So I've been doing this since before the hardware existed, and as the hardware has come, now we're getting to a better understanding. But knowing that, like with the ExoView, that you have 63, 9, and 81 on the surface and Alex inside, that still doesn't tell you if it's a therapeutic exosome or if it has any interesting content, you know. So, yeah, do we really want to get to that understanding or not? I mean, we just got to single cells a few years ago, right? I don't know if we're going to ever get to single exosomes.
And the last question I have for you is targeting. Do you have a technology which is proven in clinic or at least studied well enough to say that this batch of exosomes, I can target it to this specific compartment in the body? Well,
that's the benefit of knowing that we need 63, 81, and 9 on the surface. You know, I saw an interesting talk once that 9 was only on younger exosomes. But if you have 63 and 81's on the cell surface too, you can make whatever you want, attach it to that 63. Now you're going to have to look at the imagenicity of that, et cetera. But it gives you that value. I mean, that's a good.
Is this something you do in your laboratory?
That's something we have going, yeah.
Okay.
For brain targeting.
Thank you.
So you got approval from the FDA.
Got approval after much consternation.
Is this for human, animal?
Human, for human IV administration for post-COVID symptoms. Now, will we actually do that trial? I'm not sure. I mean, because I put it in when COVID was a very big issue. And then actually, they said to me, our risk-reward calculus has changed. I mean, they don't want any more COVID issues over there. And that's why it was so hard. I had to do a type A meeting. I had to do all kinds of things. And make a lot of data. I had to characterize the cell line the same way as if it were cellular therapy. And our argument was, we're not even using the cells. We're using the exosomes. We characterize the exosomes. No, you're going to have to do both, down to the wire, down everything that you could possibly do. Yeah.
Any questions for Duncan from the audience?
Oh, I also went through a 2-week inspection. That was fun.
I'm sorry?
2-week GMP inspection. Yeah, that was awful.
Thank you so much. Appreciate it. Hello, Ian.
This is for Dr. Ross. So just any comments you'd like to share about organoid models or any ways to sort of compare all these biologics that are emerging in a meaningful way?
Yeah, I love fibrin. The idea of, I really am interested in, what you should think of is that nano-encapsulation or encapsulation in a diabetic model of islet cells. I think that's so cool. Keep the immune system away from it, you know. So you could think of using those, putting those islet cells in that matrix and then encapsulating it away to keep them as third parties away from the immune system. That's a fantastic method. And you could think through that to other organoids such as kidney, et cetera. And then you could get away from a lot of these HLA problems that everyone here is successfully working on, but those are very difficult methods. Yeah.
I have a quick question for Duncan Ross on the exosomes. What's the difference between exosomes derived from BMAC, Wharton's jelly, and yours is placental derived? So that's amniotic fluid? Or?
Placental cell line. No, I have to apologize to everybody because I am part of this group that went to the clinic before they went to research. All right. So what has happened is that everything is being called an exosome. So I used to make an amniotic fluid exosome, and you could see some markers like CD81, but when we ran the RNA concentration, there was no RNA in it. There was very, very little. So when you see somebody says a Wharton's jelly exosome, that means that they're taking an umbilical cord, grinding it up, filtering it, and selling that within 24 hours. I was actually going to give it away for free as a loss leader because it's so cheap and easy to make, and there's no characterization. Now you could have the Ebola virus in that. You have no idea. So I was never really too chuffed about that idea. If it was an umbilical cord MSC, they would state that, meaning that they're expanding it. And you can't really call something an MSC until you expand it because of the cell surface markers and the requirement to differentiate it to cartilage, adipose, and bone. So those are the dog whistle of where it came from. Is it a tissue product or is it a cell therapy product? And mine is a cell product, and I usually have that slide up. Do not call this a tissue product, because if you say that in Europe, you are never, ever going to get approved. And in fact, I had 3 regulatory people refuse to even work on the regulatory because I didn't know how to successfully get that idea through their head. So that is a pretty key indicator there. But when we looked at all of those tissue products, not a single one had RNA in it. So there's something about this culture system that is creating this RNA. And
How sensitive is it to passage number?
it's a little sensitive, but we're using max P4, and using as long as you don't use serum, you can do pretty well. Yeah.
Anybody else? Okay, not right now.
Okay,
Dr.
I can go
Devakumar.
ahead. So the next questions I'm going to ask would be for the reprogramming people. So if both of you can answer the question Mike and Ellen. So, what needs to happen from a regulatory perspective to actually take the reprogramming technologies that are emerging to the clinic?
Hold the microphone like this. Dr. Ross gets an A+ for this. Hold it like this. This is how you hold the microphone when you're going to talk. Thank you.
I know nothing about the clinic. I'm sorry.
You don't want to answer the question?
About the clinic?
Yeah.
I don't. We're not near the clinic.
She said they're not near the clinic. What I describe, how's this? Is this good? I think it's that you don't want to hear your voice reverberating back at you like a rock star up here. So, what I described is not near the clinic. There are some people working on partial reprogramming technologies utilizing different methods, right? mRNA, there's been discussion about exosomes or extracellular vesicles to deliver gene therapy, which has really been our primary focus, and or other. I cut you in a small molecule drugs as a cocktail to accomplish that. Each would have their own regulatory pathway. Some people in the field, I won't name names here, but some people in the field have said we're 2 years away from an IND. I think that's a bit optimistic. I purposely laid out sort of a transparent and candid view of what we're dealing with here. Reprogramming, like iPS cell reprogramming factors, can take a skin cell 180 degrees across the globe, back down to Singapore, back to the beginning of life. That's a really significant alteration in the phenotype of a cell. Now, okay, we're saying partially reprogram. We're just transiently expressing factors capable of reprogramming cells back to pluripotency or a subset of those factors, but still, what I tried to allude to is these factors are the regenerative state is, I think, not purposefully, has been selected by evolution and by natural selection. I'm not trying to get teleological here. It was selected for as a tumor suppressive mechanism. And so FDA, rightly so, I hope, is going to be all over the technology about safety. Ellen's approach is, I think, far less problematic. Maybe you would disagree, but I think the approach she's taking is has less ideological baggage carried with it. But partial reprogramming, I've been as enthusiastic as I can be about the potential of the field and where it's going to go. We've got a lot of work to do. But the safety concerns are going to necessarily have to be addressed in depth. And there's a lot of work to be done. But the potential is, I was sitting here as the panel was sitting down, and I was thinking, what I would like to talk about. I won't try to change the topic of Q&A, but I really feel strongly that those of you that are here today, we all have a common interest in advancing this field, and the problem the United States and other countries are facing with what I talked about, the chronic degenerative diseases, is going to bust the bank. Duncan, well-known economist and financial guy, just gave a talk at UC Irvine, I think, with a very candid appraisal of where the United States is headed. And he came up with this number of $200 trillion net present value of our obligations under our entitlement programs to the aging population. What is that? $200 million?
Oh, you're looking at me.
You're the intellectual here in the panel. You can do the math.
A lot. It's a lot.
Right? $200 million. What is it, 300 plus million in the United States? That's almost $1 million a person. and his point was, well, we're not going to meet that need. The United States can't do it. It's not even budgeted. Zero is budgeted for this. So I really would like all of us to try to impact as much as we can policymakers in the United States and around the world to take a hard look at the technologies we're talking about in this conference. It's not just a matter of health for the individual and the suffering of individuals, it's also the fate of our countries. It's a very serious issue. I wish we could get the attention of people on it. So sorry to get on my soapbox, but I just wanted to say that.
No, that's absolutely relevant. Thank you for that, Mike. That's absolutely relevant. Thank you for that, Mike. So, Ellen, there are a lot of people working on trying to reprogram cells and tissues using Yamanaka factors. There have been approaches using chemical means to reprogramming cells, and your approach is also, the data that you presented was phenomenal. So why aren't too many labs working on similar approaches such as yours? Why is there such a mad rush to use Yamanaka factors and reprogram cells? And of course, Mike rightly pointed out all the problems that could exist in that pathway.
Well, I guess to be cynical, it was published in Nature. But, well, I mean, we found, again, through this mouse that has really incredible regenerative properties. I mean, besides the things I talked about, I really never talked about optic nerve. I mean, we could regrow the optic nerve after a complete transection in the MRL. And we have spinal cord data where we can regrow the spinal cord. I mean, it's actually amazing. So that we've found the molecule was really, based on the fact that we went to this DARPA meeting and about mitochondria, and so why aren't people looking at this? I don't know. I don't know. I think that the Yamanaka factors can. I think that HIF, or maybe a combination of things. Maybe it's more than HIF. But I think that it could be a transient. In a way, it's nice because it's a transient response that comes and goes. And I think that that's important. If we give our drug in over time, it doesn't work. It has to be a very short treatment, and I think that there is this dedifferentiation step, and then it goes to another state. We haven't really looked at that. But, and on the other hand, if you do transfection using these 4 factors, or how many, there are, you don't really have the ability to sort of be nimble with them. They get turned on, and you can, of course, turn them off. But I think that that's actually a key issue, and I think that that's actually important in what we're seeing. So you hit it early, you turn up the HIF levels, and then it comes down. I mean, this is what happens in the MRL mouse. It goes up, it comes down, and then does whatever it does. But why aren't people using HIF or other things? I don't know.
I can't add anything to what Ellen said.
Vijay. That was a phenomenal presentation. And I don't really know what to ask, what questions to ask of that presentation. So let me ask the audience if they have any questions for Vijay.
Yeah, so not hard to imagine the need for LIMS, right, and other VCAs. And your presentation was very overarching. You talked about treatments, you talked about diagnostics, you talked about surgery. As you think about putting this into the clinic, which one of those problems is going to be the hardest to solve, and why?
I think any problem has to be addressed in context. I think, and also the timing of the problem as well. I mean, you've had lots of discussion happening here about aging and reversal and reprogramming and all that. It's hard to take something you do as an experiment in the lab and put it to the real world because there's so many things that confound real world applications, right? Let's say optic nerve injury. I mean, when we are dealing with optic nerve injury, you don't have a patient coming to you immediately after injury. They come sometimes months later or years later, sometimes acutely. So you can't control these things. And every patient is different because immunologically, the homeostasis, the milieu, everything is different. And the wound, for example, if you talk about injury, you can't talk about it in its purest sense because there's contamination in some, there's no contamination in some, there's age, there's other factors that come into play. So science is beautiful when you can control it. The results are also beautiful, right? But when you really put it to your real world use, it's difficult. And your question is difficult in that sense because it's hard for us to expect, I mean, obviously, if I am being asked, okay, what are the diseases that you need to treat? I mean, all of these are important. I think blindness is a very big deal. I mean, nobody wants to be blind, and there's lots of vision issues that don't need to be traumatic. There's progressive visual diseases that happen that can be slowed, right? And that could be extremely important as well, like preservation of vision, even though it's like not 20/20, is very important, right? On the other hand, when you ask me as a reconstructive surgeon, where this is going to head, anything that we could make safer would eventually revolutionize our practice because we can go into concierge reconstructive surgery. We can take noses and treat, transplant noses or ears. The noses and ears are extremely difficult to reconstruct, almost impossible. Lips are next. Eyelids are next. So we could talk about those reconstructions. You could change lives, and you could do joint replacements, just the whole joints for chronic diseases. Obviously, if epigenetics becomes reality, you may not have osteoarthritis or rheumatoid arthritis or chronic diseases. But until then, these become potentially applicable. So yeah, I mean, I would probably, plastic surgery itself, like aesthetic surgery, may completely become extinct when, let's say, Dr. Ross comes up with these exosomes, which you can inject with microneedles and not have any aging or whatever. I mean, like, cosmetic surgery is going to change, right? I mean, you can have aestheticians do this in the clinics. Right now, we have those things happen, lasers and chemical peels and other things. So these technologies are changing practice of medicine. They're changing specializations. They're changing us as practitioners. So I hope I didn't give you a long-winded answer, but yeah, that's my answer.
Yeah, I have I have a partial answer to the question you asked. First of all, in full disclosure, Ellen Heber-Katz is my wife, so maybe there's a little bias here. But you asked the question, well, why are more people not taking this drug approach? Well, if you take a look at the title of this meeting, it's the World Stem Cell Summit. And there has been a confluence of the concept that regeneration is stem cells, or stem cells is regeneration. Certainly stem cells are a component of regeneration, okay? But they aren't regeneration. So people, when you think in terms of transplanting cells, right, you don't think in terms of drugs, okay? The possibility of inducing an entire regenerative state by upregulating a molecule HIF-1A is a purely pharmaceutical approach. It has literally nothing to do with stem cells, but it is quite potent regeneration. Although the stem cell phenotype does come up. It is, as you saw.
Sure. So I clearly understand what you're saying, but Stem Cell Summit started 15 years ago. Bernie has been doing these conferences for a very long time, and now we are actually moving from stem cells to all other technologies that can produce or effective rejuvenation in the body. That's why, if you see the last couple of days, the talks, there were hardly any stem cell talks. They've mostly been on different technologies, all of which produce rejuvenation in the body. So, Vijay,
can I add a comment to that really quick? Here.
Oh, okay. So,
I just want to add to this that I still think, and this present meeting excluded, I still think there's not enough interaction between the aging biology field, the stem cell field. I mean, MSCs is a great example. There are MSCs being injected in people all over the world. I've had IV MSCs. I'm alive still, so that's good. But we really have the aging biology field has largely ignored stem cells, and regeneration is a way to sort of bring those two things together, I guess. But going back and asking questions like, do exosomes from MSCs affect biologic clocks of aging? Do MSCs themselves affect that? Or other tissues, other cell sources. These are fundamental questions, and I think we really need to work together to try to achieve some of the answers on that. And I'm positive about these approaches. I think they're likely to work, but it's hard for me to point to real data, at least on aging biology, that's quantitative and controlled to make that case. And I think that we need to work more together to try to achieve that, because I think we're missing opportunities here.
Absolutely. So, for example, we have two different approaches for reprogramming here. And yesterday we had a presenter from University of San Diego. She spoke about plasticity based with the lymphatic support as to how stem cells from the intestine can behave very similar to the stem cells from skin and vice versa, and the effect that the lymphatic system has on the plasticity. So I think we have different approaches. If we combine all of them, we will have a much better solution. So extending the same thought, which I do have a question, we briefly discussed that offline, which is immunomodulation of MSCs and its applicability to all the transplant work that you're doing, immunosuppression work that you're doing. So I thought maybe the audience also should hear your comments on that.
Yeah, I mean, when you look at MSCs as a science, they've been around for a while, and there's a lot of loose terminology applied to MSCs, as you know. I mean, there's adipose-derived, there's bone marrow-derived, and there's different origins of stem cells. There's autologous stem cells, there's transplanted stem cells. The main reason why lots of this work has not come into transplantation is because it's extremely difficult from a regulatory perspective, because you stand in front of these FDA regulators, and they ask where are these cells going, how many cells do you need if you have the same disease across different.
Sorry, the question I had was immunotolerance induced by MSCs.
Yeah, that's what I'm coming to. So, there's you have concepts like immunomodulation, immunoregulation, tolerance. I mean, all of these are on a spectrum, and they mean different things to different people. Although there are definitions out there, the way you use these definitions it has become very difficult. Obviously, you go to. I mean, I just came from a transplant congress in San Diego. We had a whole tolerance session, right? I mean, what is tolerance? Is it complete? Is it something that you define as a state that is free from immunosuppression? Maybe. Some people say something about probe tolerance, like, is somewhere in the middle that can be abrogated by infection or a little bit of rejection trigger. Now, MSCs have shown promise, right? But then the replication of these studies between different populations has become very challenging. Like, even if you standardize the dose, the timing, the repetition of these doses, it has become very difficult. I mean, there are some successful studies, but the generalizability of those studies has become very difficult. Now, MSCs have one of the possibilities which is really showing promise is exosomes, because you potentially can eliminate the disadvantages of stem cells by themselves, which is the viability issue, which is the propagation issue, which is the scale-up issue, and then look at potentially, hey, can you get the benefits of the growth factors or the pluripotent, like the paracrine efficacy or the pericyte stabilization, whatever that might be, by using an exosome pathway. And then exosomes becomes even more interesting because there can be more specific aspects of exosomes derived from regulatory cells or derived from dendritic cells or derived from so there are companies out there that are doing regulatory cell exosomes, and we are looking at it as we speak in some studies which are showing very good promise, because you can decorate these exosomes with drugs, you can pegylate them, you can add immunosuppressive drugs, you can encapsulate them. There's potential for transplant with cell-free therapies, if you want to call them, but they originate in cells, but they're cell-free. Maybe that'll become more of a mainstream, because regulatory burdens or barriers will become much more easily, broken down. I don't know if I answered your question.
We have a question right here. Right here.
Hi. I'm curious about the conversion of nanotechnology with regenerative medicine. As an example, would it be possible to have nanorobotics monitor some of these transplants and be able to get back that data to see if that particular organ is efficacious and is working within the body? Am I out on the left field on that, or is that a possible convergence? Because if I implant an organ, how would I know throughout time if it's working if I don't have real-time data or real-time monitoring of that? And if that's the case, would that data be useful to the originator of those organs or products?
This question is to the panel, or this question to a particular person? Okay. I can take you want to have a shot?
No, I absolutely agree. You have to have some monitoring. Address that.
Yes. So there is a lot of technology interest and advancement in the area of nanobots or micro robots that can be injected, like in capsules, or potentially as chips, as nano carriers. There's now ingestible electronics, which are electronics that you can ingest and then they get completely degraded after time. They're being used in GI diseases like ulcerative colitis or even small bowel transplants, some of which has been published. We are working with a group out of ETH in Zurich, as well as in Northwestern University. That's a field called integrated bioelectronics.